Hello guys, my name is Dr. Varun and here I am back with 30 days, 30 most important topic for NEET 2025 biology.
Chalo, let's complete it.
Well, in today's video, we're gonna be covering about two most important topic of chapter photosynthesis in plant. What are they? Number one is the Chemiosmotic Hypothesis. Yes, I know that you all are struggling with it. I'm gonna help you out with the easiest thing and quick thing about it. Then coming up to the next thing is the dark reaction, that is biosynthetic phase, about C3 and C4 pathway. Let's just dive into it. So starting with the Chemiosmotic Hypothesis. First of all, what is Chemiosmotic Hypothesis? Chemiosmotic Hypothesis. Now remember one thing. What is the role of Chemiosmotic Hypothesis? This is what we are making ATP. Okay, ATP is what? Chemicals. So basically what? We are making ATP. Osmosis means going things from one concentration to other concentration. What? Higher concentration to lower concentration. So movement of thing, what? Movement of which chemical? H positive means protons from higher towards lower. Something is doing it. And hypothesis means we, like the person who gave this thing, he told that okay, this happens like this, then only the ATP is being produced. So this is the major thing. So here on the basic, just remember that one side we will increase the concentration of H positive. Okay, if you remember the ETS, in ETS also what we are doing, we are removing H positive from one side and moving it to the another side, and where the side the H positive is finally going, it is giving us what? With the help of ATP is, okay, it will give us what? ATP. If you remember that, if not, then go back and see the yesterday's video. Same thing is occurring over here and same person only gave it. The only difference is the places where it has given. This, where it is occurring inside the chloroplast. Where inside the chloroplast? The inside structure is the thylakoid lumen and the outside area is the stroma. In between them the membrane that would be present, that is what? Thylakoid membrane. Okay, so this thing is there. We have talked about a lot of things. Let's just see about it. So imagine that this is your chloroplast, and inside chloroplast, this is what? This is the stroma area. And these are what? These individuals are the thylakoid. So just remember this thing. So I am what? I am enhancing this area over here to you. Okay. So what is present? Over here we have a thylakoid membrane. So we are making the thylakoid membrane on the outer side, inner membrane and the outer membrane. Okay, so this is what your thylakoid membrane, making it little. So in this, I am just telling you the things. So this is what? This is the membranous space where all what? Your photosystems present, and here only what? Your light reactions taking place. During light reaction, certain things which are occurring, which is doing what? Which is increasing the concentration on one side and decreasing the concentration on the other side. Where the concentration is increasing? Inside this area. This area is called as what? Lumen. And where the concentration is decreasing? In the stroma. So means this area it is decreasing. Inside the lumen area it is increasing. So here the concentration decreasing. Of what concentration? H positive. Here it is increasing. So let's just see that what is there at how it is decreasing. So at the two places where it is decreasing, two places it is increasing. Okay. So this is your membrane. I am making it little bigger. Imagine that this is the membrane. Membrane. Okay. So this area would be what? This would be the lumen. Okay, and this is the membrane area. Now in this membrane, photosystems are present. So if you remember about the photosystem, what we have done, that there is what? Initiation by the photosystem 2. Photosystem 2 will do what? It will excite the electrons. The electron will go to the primary acceptor. It will give the electron to the next Okay, so next electron carrier, finally it's going to the next electron carrier, and then it will go to the finally to what? Finally it will be goes to the photosystem 1. Simultaneously photosystem 1 will also get excited and it will release 2 electrons. It will goes to what? It will goes to the your acceptor. Then acceptor give it to the FED finally structure, and it gives to what? It gives this thing to the NAD positive forming NADPH. This is being formed. Now let's just see that what is happening. So remember that at the two places H positive is increasing and two ways H positive concentration is decreasing. Okay, we know that till now the concentration inside lumen will be increasing, outside in the stroma it will be decreasing. So first let's see that how it is decreasing. So decreasing from the stroma, when this reaction is taking place, this carrier 1 to 1, it do not go by the negative charge. Protons are needed, positive charge is needed to balance it. I will remember it like that. So what will happen? From the stroma, it goes inside. So what happened? That your stroma took up away the H positive and throws it into inside it. Means first H positive goes to the carrier, then carrier give it to the next carrier, the hydrogen goes inside. So this how what happened? Outside it get reduced, inside it get increased. So first way is through this way. So A option is this. Okay, then comes the second option. Second way, how it is getting increase inside. Okay, because we are studying about the increment. So the second way of increment is that since once the electron is excited from this whole structure, now this requires electron, continuously electron, or else there will be photo-oxidation of the chlorophyll that will occur. We don't want that to occur. So what will happen? Water will break down over here. So H2O will break down into H positive, electrons plus O2. The electrons will be given to this oxygen, will be evolved out. H positive will be thrown inside. Second way, in the lumen it is increasing. So this is on the lumen, this is on the stroma side. Now again you will ask like, okay, this we understood. What is the second way of decrement of the H positive inside, like out in the stroma? Then in the stroma, do you see the last step where NADP positive is changed to NADPH? Means how it will be changing it? When the H positive coming up, where it is coming up? It is coming from the stroma. So this reaction is occurring in the stroma. So second thing is from the NADPH formation. Okay, so this you have to remember. This way now what problem occur? The outside in the stroma, H positive decrease, inside it increases. Now we have a membrane. On this membrane there is a presence of what? There is a presence of F1 particle. So what happened? This, sorry, I made it a little wrong. This F1 particle would be like this. That F0 will be embedded, F1 will be outside over the stroma, because from higher to the lower it will go. Higher concentration is what? In the lumen. So everything from lumen, it will go from here to here. So as it is going from here to here, what will happen? Again the outside it will become high, inside become again low. Continuously this will happening. Along with that what? ATP will be keep on forming. I hope you would have understand that how chemiosmotic hypothesis is working. At the end, they have even said that to start this ATPase pump, there is requirements of ATP. So what happened over here? Certain channels are present, receptors or gates are present through which some of the H positive from stroma goes inside and makes ATP here, which will start this process to occur. So this thing only is the major confusion of the student, like what is happening over here. But I told you to start the system, it requires ATP. Now the ATP is not present on this side, lumen side. How it will present? If H positive will go inside. So certain H positive for the initiation of the process it goes so that it can switch on. Once switch on, no one is going there. It is making ATP, ATP, ATP. Well, that's about the chemoosmotic hypothesis. I hope you would have understood. Next topic is your C3 pathway. So in C3 pathway, what happened? First of all, for each cycle that we're gonna see, it gonna fix only one carbon. One carbon means what? The glucose is this compound. It has what? 6 carbon. When one cycle will run, it will fix only one carbon. So this 6 such cycle we have to run to complete one molecule of glucose, and from here the question will come. So if someone will ask in C3 cycle, how much ATP, because this is the dark reactions. Dark reactions require what? ATP. So there should be certain trick to remember. So first you will remember the trick, then we will go into deeper. So the trick of the reaction is 3 to 1. What is 3 to 1? 1 means for fixing one carbon dioxide, we requires 2 NADPH and 3 ATP for fixing one carbon dioxide. Means in 1 Kelvin cycle, this much will be required. So if someone will ask, right, to make one molecule of glucose, one molecule of glucose means, it means that 6 carbons will be present. So means 6 carbon cycle will be going to take place. So 2 into 6, that is 12 NADPH will be required. Whereas 3 into 6, that is 18 ATP will be required. This is the first you have to remember the term how to do it. Moving on to the reactions. So the reaction is that all the questions will be covered over here. This thing is occurring into what? In the mesophyll cells. So in mesophyll cells, what happened? Carbon dioxide come, one carbon dioxide molecule come, and who will accept it? It will be accepted by Ruby. Ruby is what? It is ribulose bisphosphate. Enzyme which is working over here is what? Rubisco. Which part of the Rubisco? Ribulose bisphosphate carboxylase, because carbon fixation occurring. So this is what? Carbon fixation step. 3 steps are there. Now in this process, what will happen? It will form certain structure. What would be the structure? It will be forming a 6 carbon compound, because Ruby is 5 carbon compound. This is 1 carbon. 6 carbon compound. This 6 carbon compound is highly unstable and it breaks down into 2,3 carbon compound. This 3 carbon compound is what? Phosphoglyceric acid or phosphoglycerate. Okay, so this phosphoglycerate, what will happen? Now this is the first step is over. So first stable compound is 3 carbon compound. So this is the stable compound, phosphoglyceric acid. This you have to remember. Now what will happen with the phosphoglyceric acid? Then the next step will start, and that step is what? That is known as reduction. Reduction means, reduction everyone know that from that redox reaction. So I'm not explaining it in deep, just remember that reduction happened. So in reduction, what happened? There is a removal of what? There is a removal of oxides. So over here also from acid, oxygen will go. Okay, so this thing will change into what? It will change into aldehyde. Okay, so as it changes into aldehyde, that aldehyde will have phosphate. This is phosphoglyceric acid. So this is a 3 carbon. This will be what? 3 carbon aldehyde, that is aldose phosphate or phosphoaldose. Now this is what? The reduction step, that is the second step. It requires what? Energy. So here the energy requirement would be there, and that energy requirement, the series of steps. Okay, so there will be 2 ATP required and 2 NADPH will be required in this reaction. We know that 3 ATP and 2 NADPH. 2 NADPH is already required. 1 ATP still left to be seen that where it is being required. Now this aldehyde, this 3 carbon, what will happen? It will forms what? It will remove 1 carbon dioxide, which will go and form what? Glucose 1 by 1. Whereas the 2 carbon compound which is left, it will join and forms what? Again the 5 carbon compound. This step, that is the third step, is known as regeneration, and during this step only there is a requirement of what? 1 ATP. So this is C3 pathway. Okay, next comes the C4 pathway. So first question, same like in C4 pathway, would be what? C4 pathway is what? It has what? C3 pathway definitely present, but certain extra steps present. And this is occurring in those plants which is in the dry arid areas, so that they can conserve maximum of their resources. Okay, now in the C4 pathway, their anatomy is different because they have thick bundle sheath cells, and inside bundle sheath cells only their C3 pathway is going on. If someone will ask like how much amount of ATP and NADPH will be required to fix one, then it increases. What it is? The 5 to 1 means for one carbon dioxide fixation, for the one cycle, in the one cycle, what will happen? 2 NADPH, which is similar like the earlier one present, but 5 ATP required. Earlier it was 3 ATP required. So to make one molecule of glucose, you will require what? 2 into 6, that is 12 NADPH, similar like that, but 5 into 6, that is 30 ATPs, which will be required during the regeneration and formation of other component. Okay, so this thing we have to remember. Let's just see that what is taking place here. Remember, here your mesophyll cells do not have the Rubisco enzyme. So Rubisco enzyme is present in the bundle sheath cells. And why they are doing it? So that they can increase the concentration of carbon dioxide. They can survive for the higher level of the carbon dioxide, which is not possible in the bundle sheath cells. Okay, so affinity, it will increases. Okay, I'm doing it little quick just to give you the glance of what is needed. In detail, we are not going that much. Okay, so there are two different types of cells. So we're gonna make the two different types of cells over here. This is your mesophyll cells. This one is your bundle sheath cell. Now between them, these are what? These are their plasmodesmata connections. These are, you know that in the plant, plasmodesmata connections are present through which things can be transferred. So this is your mesophyll cell. This is your bundle sheath cell. Now you have to remember the facts that in the mesophyll cells there is a presence of what? There is not presence of Rubisco enzyme. So cut over here. Whereas over here Rubisco enzyme present. Okay. Now the next thing is what reactions are taking place. So carbon dioxide will first come on this cell. Okay, that is a mesophyll cells, and as it goes inside, it will join with what? It will join with, like, of course, first it will join with the water and all, which is given in NCERT, but the main thing is the enzyme which is joining it with. It is joining with a phosphoenol pyruvate, that is a 3 carbon compound. So 3 plus 1 carbon forms how many carbon compound? 4 carbon compound. And this 4 carbon compound name is oxaloacetic acid. Okay, this oxaloacetic acid, this first step is the fixation, carbon fixation. Okay, then this will change into what? It will change into malic acid. It can even be changed into aspartic acid. Okay, then it can transfer to the next cell, that is bundle sheath cells. As it will reach to the bundle sheath cell, what will happen? Your 1 carbon dioxide will be removed and a 3 carbon compound again will form. This 3 carbon compound will go back, because it won't stay here in the bundle sheath cell. It will go back again to the mesophyll cells, where it will regenerate itself into what? PEP. Now, what happened to this carbon dioxide which is coming here? It will undergo Calvin cycle, same cycle, and forms the food, because this contains the Rubisco. So Rubisco will undergo the Calvin cycle once the carbon dioxide came. So carbon dioxide keep on coming like that. Major question over here would be that first thing is that over there, this reaction, which enzyme is doing it? So the enzyme name is pepcase. This is the extra thing. acceptor is 2C, for first stable product is 4C, that thing also you have to remember. And here during the regeneration, extra ATP that is required will be used up, and some will be used up over here. Okay, so this thing you have to remember about it. Well, that's all for this video. I hope you would have understand the crux and the quick revision of the things. We are not going in very deep, but this is the major crux that you should remember. If you like this video, give it a like, share it with your friends, or maybe not, as I usually say, because it's time to be selfish. But I hope you like this video. Don't forget to subscribe if you haven't. You can follow me on my WhatsApp channel, that is Biology Simplified English. Well, all the very best, and we are keep on coming up with the more things. Moreover, tomorrow I will be releasing a question paper which will be of 90 questions for biology. Please do that paper. It will be available as a link in the description also tomorrow's videos, or it will be available on my WhatsApp channel as well. Do that paper. For that paper, whosoever want it, they can send it to me. I will analyze and help you out finding your weaknesses, so that we will get 360 out of 360 for sure. Well, that's all for this video. Thank you.